Okay. Hello, everybody, and welcome to Beyond Genes. I'm very, very sorry about the delay this morning. We had some technical problems. So without further ado, I'm going to run through the intro and turn it over to our keynote, who is Dr. David DeMarini. My name is Jill Escher, and I'm your conference chair. I want to thank our sponsors. I won't list them all in the interest of time, but they're wonderful. Thank you for contributing your brains and some funds. I want to thank our committee. I think that everybody put together a wonderful program. And this program exists in 6 parts. Today is the intro and mechanisms part 1 with some senior researchers and we have more presentations by senior researchers on October 21st, and then some talks by some young investigators, November 6th. And then we switch gears and turn to some exposures for which there are very good data indicating heritable effects through germline. And the basic theme of this conference is that heritability is greater than just DNA. Logistics here, we ask you not to record any talks, although many of the talks will be recorded and uploaded to our site, so please stand by. We anticipate maybe about half of the talks throughout the conference will end up being recorded. Each talk will be followed by a Q&A session, so please enter that into the question bar. Moving forward to Dr. DeMarini. We're so pleased to have Dr. DeMarini open this conference. For 35 years, he was a genetic toxicologist at the US EPA, where he helped determine the mutations induced by drinking water, air pollution, and other complex environmental mixtures. He is past president of the Environmental Mutagenicity and Genomics Society, and has worked extensively with international agencies to assess the mutagenicity and carcinogenicity to humans of environmental chemicals. And I know that he has a very keen interest in what may happen to germ cells. He's written about that several times. So with that, I'm going to turn it over to David.
All right, so the title of the talk is Germ Cell Exposures and Heritable Effects. Is our regulatory paradigm failing to protect future generations? So here's a brief outline of the talk. We'll start about 100 years ago with the discovery of germ cell mutagens. Then we'll go back about 50 years ago to the founding of the Environmental Mutagens Society. Then we'll go to the present to discuss what we know about germ cell mutagens in humans and in rodents. And then we'll discuss current germ cell testing requirements and whether the testing paradigm should change or not. So let's see. First of all, I have to get rid of this off of my screen. Let's see. There we go. So let me get this up here. There we go. So first of all, we need to be on the same page, which I'm sure we are, which is that germ cells are not somatic cells. They are very different, completely different genetic processes, completely different epigenetic processes. They are different in males and in females, which is generally not the case for somatic cells. Obviously, they're dependent on signals from adjacent support cells. This is also true of some somatic cells, but profoundly true for germ cells. And of course, there's minor defects in germ cells can blueprint major defects in offspring development. So the point here is that they are not the same. And so the question is mutagenicity testing primarily, almost exclusively, only in somatic cells going to provide protective information for germ cells? And I think, as you will see later, the answer is no. Well, in one slide, let me briefly go through the history of germ cell mutagenicity, which really started nearly 100 years ago in the 1920s with the discovery that X-rays and UV light were germ cell mutagens in fruit flies, corn, and barley. Of course, this is the work of Herman Muller at the University of Texas at Austin, for which he won the Nobel Prize. Lou Stadler at the University of Missouri in Columbia, and Edgar Altenberg at Rice University in Houston. It's very interesting. All this early work happened in Texas and in Missouri, of all places. About 10 years later, in the mid-1930s, all of this work moved finally into mice, where X-rays was shown to be a germ cell mutagen. But this was really chromosomal effects. And this was in the mouse dominant lethal assay developed by Snell here in the United States and Hertwig and Brennick in Germany. During World War II, of course, Charlotte Auerbach, and shortly after the war, published on the first chemical mutagen, germ cell mutagens. These were mustard gas and mustard oil in the fruit flies. And of course, Dr. Auerbach was not the only one doing this. Right after the war, other people like Joseph Rappaport in the Soviet Union, many others in Europe, also started showing that various chemicals were indeed germ cell mutagens in fruit flies. All of this really came to a head in 1951 when Bill Russell invented the first germ cell gene mutation assay in mice at the Oak Ridge National Laboratory in Oak Ridge, Tennessee, and showed that X-rays was indeed a germ cell mutagen in mice for gene mutation. And that really broke the whole field open because everyone agreed mice and men were not too different. And if you could see X-rays being a germ cell mutagen in the rodent, it very likely was so in humans. So all through the 50s, of course, after the war, tons of new synthetic chemicals were introduced into the environment. None of them tested, of course. There was no EPA. And this raised some alarm bells for Josh Lederberg, then at the University of Wisconsin, that there was the possibility of a lot of germ cell mutagens being introduced into the environment. And he brought this issue to the attention of the National Academy of Sciences. Lederberg, of course, eventually won the Nobel Prize and was one of the founding members of the Environmental Mutagen Society. In '68, James Crow, also at Wisconsin, published a report of an NIH germ cell mutation conference that he had organized. And I mention this because that report was seminal for the founding of the Environmental Mutagen Society, and most critically, for the initiation of germ cell and mutagenicity testing of products before they were released to the public and for drugs. So it's a very important development. At the same time, Sam Epstein at Harvard showed that some environmental chemicals were also germ cell mutagens in the mouse dominant lethal assay in mice. He published that paper in Nature, got a lot of attention. And the next year, Epstein and Marvin Legator at the time was at the US FDA, proposed for the first time, it was in a government report to the Department of Health and Human Health and Welfare, that pesticides should be tested for mutagenicity. And this was the first time that mutagenicity testing, was proposed within a government agency. And at that same time, the EMGS was founded to eliminate germ cell mutagens. And also, of course, the agency or the organization expressed concern about the intellectual and physical health of future generations if such agents as germ cell mutagens were being introduced into the environment. I've reviewed all of this in a very recent paper, which you can go see. Well, let's flash forward 50 years and ask, well, what are the rodent germ cell mutagens? Well, there are 85 of them that are well known. Most are cancer chemotherapeutic agents, and this is because these are drugs that are actually given to humans. They're very potent alkylating agents. They were well known to be somatic cell mutagens to begin with. But there are, in fact, a number of environmental germ cell mutagens that are known in rodents. And this is side stream, mainstream cigarette smoke, the particulate fraction of outdoor air pollution, and of course, diesel exhaust. And all of these are very important. And every single one of you listening to this are exposed daily, chronically, day and night, all of your life to the particulate fraction of outdoor air pollution and to some extent to diesel exhaust. So these are not exotic agents. All of us are exposed to these, and these are well-known rodent germ cell mutagens. And Francesco Marchetti and his colleagues at the University at Health Canada in Ottawa have reviewed all of this very recently. Well, that's the story in rodents. What's the story in humans? Well, that's easy. It's zero. There are no declared human germ cell mutagens, and we're going to discuss why in just a second. Also, I should note that there's no absolute, and I do emphasize the word absolute requirement, for testing for germ cell effects by EPA, FDA, or any of the other international regulatory agencies, and I'll discuss that in a moment too. Well, how did germ cells fall off the radar? Well, as I showed you earlier, the real focus was after the dropping of the atomic bombs in 1945, the concern really was for the potential ionizing radiation via germ cell mutagen. But all of that shifted in the mid-70s with the development of the Ames assay as a screen for detecting potential human carcinogens and for Richard Nixon's war on cancer. And consequently, all the research funding, the regulatory requirements, and all the research efforts began to shift from germ cell mutagenesis to detecting chemical carcinogens as mutagens. So this was a really profound shift in everything. There also, of course, was a failure to appreciate epigenetic germline effects. And our next speaker will discuss this. He's one of the founding scientists in this field. And so that also was not on the radar. Perhaps most pernicious in all of this is the assumption to this very minute by essentially all clinicians that de novo mutations are simply random. These are just errors of DNA metabolism. And so that when a clinician sees a child with a mutation that is not present in the mother or the father, the notion is that mutation just happened by chance. It was not it did not happen due to the exposure, for example, of the father to some environmental agent causing a germ cell mutation in him, which was then transmitted on to the child. And there's reasons why clinicians have been loath to accept this notion that there could be an actual cause for these de novo mutations. So I'm going to discuss that. And so what are those reasons? Well, one is there's never been an agency to declare anything to be a human germ cell mutagen other than the Atomic Bomb Survivors Commission, now 70 years old, established after the bombs were dropped in 1945. And obviously their mandate is only to declare ionizing radiation from those two bombs as germ cell mutagens in the survivors. So, and they have not been able to make such a declaration, by the way, in 70 years. And so it's a very narrow mandate. And so that's one of the reasons. There's no one around to make such a declaration. This was also probably the wrong population to study. The bomb survivors largely received an acute, not a chronic exposure. We now understand a chronic exposure is really what's probably required. They've looked exclusively at coding, not non-coding genes, and at single gene versus complex multigenic traits. So this is missing a lot of potential activity. It's the wrong tools. They have not applied next-gen sequencing or gene expression analysis. It is difficult to study human female germ cells and early stage male germ cells, but those are the stages that we now appreciate are really the ones most vulnerable. Finally, I should note that regulatory agencies do not regulate based on mutagenicity. We have known since the 70s that air pollution and cigarette smoke are somatic cell mutations, and more recently that they are both germ cell mutations, all in rodents, but none of them are regulated based on that. Air pollution is regulated based on mortality, how many people die when the air pollution gets to a certain level, and smoking is regulated based on lung cancer. So there is no tradition, there is no precedent in the world by anybody anywhere to regulate anything based on mutagenicity, let alone germ cell mutagenicity. So for these and other reasons, there is just no acceptance or sense by clinicians that exposure of potentially fathers to potential germ cell mutagens might in fact be causing some of the mutations they see in children. Well, a quarter of a century ago, my former boss, Mike Shelby, who still lives here in Chapel Hill, just down the street from me, published a very important paper in 1994, and in 2012, I expanded on it. Mike was at NIHS at the time, and the question he asked is, really, are there no human germ cell mutagens? Can this really be true? And he put together this very informative table, and on the left are a series of mutagens. The ones in white are the ones he had included, the ones in yellow are the ones I included, although he also did include ionizing radiation. And what you can see is that in rodents, all of these are carcinogenic, all of these are somatic cell mutagens, and all of these are germ cell mutagens. And in humans, they are all carcinogenic. Essentially, all of them are somatic cell mutagens, and yet we are forced to say that we don't know whether any of these are germ cell mutagens in humans. Really? As I showed you the paper by Marchetti et al. From 2020, in that paper, Francesco and his colleagues have reviewed the biology of germ cells in rodents and humans, and they show that they are essentially identical. So there really is no biological basis for assuming that for some strange reason, agents that are germ cell mutagens in rodents would not be germ cell mutagens in humans given an appropriate exposure to the human. So these are probably all pluses, but why are they question marks? And the reason is for the reasons I just showed you. There's no commission or international body to make such a declaration. And today, unfortunately, or fortunately, the bar has been raised very high such that the basis for such a declaration would rest on next-gen sequencing, seeing a de novo mutation in the child that's not present in the parents, and having some epidemiology linking the exposure of probably the father prior to conceiving the child to some environmental agent or drug resulting in the mutation. So it's a very high bar, and so we just simply don't have the answer. But most of us in this field view that, of course, there are human germ cell mutagens. It's just a matter of getting the right evaluation. So I tried to devise the right evaluation. In 2012, I came up with the first really systematic approach to trying to determine what would be the criteria and the process by which you would declare something to be a human germ cell mutagen. And I based it on the now over 50-year process that we all accept, which is IARC's evaluation for what is a human carcinogen. IARC is the International Agency for Research on Cancer. It's in Lyon, France. It's part of WHO. It's highly respected. It is what decides what is and is not a human carcinogen. And using those criteria, but changing them from carcinogenicity to germ cell mutagenicity, we put the animal evidence together for these various agents, ionizing radiation, chemotherapy, smoking, and air pollution, the mechanistic evidence in humans, and you come up with a positive evaluation. So I'm completely convinced, as are a number of other people, that at a minimum, these agents are very likely human germ cell mutagens. It's just a matter of having an international body to agree on that and assessing the evidence in this manner. Just to take it one step further, a couple of years ago, Mark Beal and Francesco Marchetti and Carol Yacht, all at Health Canada in Ottawa, published this very important paper where they pulled together all the literature on cigarette smoke as a human germ cell mutagen. And I won't go through the slide, but it is extensive and very compelling. And I think actually today, the best data now really are for cigarette smoking and certainly for ionizing radiation as likely human germ cell mutagens. Well, let me spend just a minute now discussing mutagenicity testing history. So again, as I noted earlier, the testing of pesticides for mutagenicity by EPA began in 1976 under TSCA, the Toxic Substances Control Act. And so, and this caused other regulatory agencies around the world to also start testing for mutagenicity of drugs and environmental chemicals before approval. EPA published its first guidelines for mutagenicity risk assessment in 1986, focusing exclusively on transmissible germ cell genetic risk. So germ cell testing has really been very, very much a part of testing procedures for a very long time, almost from the very beginning. There were efforts in the 1990s to classify agents as human mutagens, but no criteria were ever set. The approach used was never wasn't systematic, and nothing ever came of it. And so 8 years ago, I proposed what I just described to you, a classification scheme and criteria by which one could do this based on IARC's carcinogen classification scheme. So I think it is doable. Well, what are the mutagenicity testings of regulatory agencies worldwide? Well, they do vary slightly, a little bit from country to country, region to region, but they are essentially 99% the same. What they involve is first, the agent is tested for gene mutation, typically in the Ames assay in bacteria. Second, it's tested for chromosomal mutation, typically the micronucleus assay in the mouse bone marrow or else in cells in culture. And thirdly, if a chemical analysis shows that the chemical in the rodent, after the rodent is exposed, that the chemical gets to the germ cells, then one could do germ cell assays, either the dominant lethal assay or gene chromosomal mutations assay in sperm. But of course, that kind of chemical analysis is rarely ever done. And consequently, germ cell assays are hardly ever done. And if so, they're done really only in males for mutation, not for gene expression, transmissible changes in gene expression, and not for phenotypic effects. And all of this has been reviewed in the papers I've cited below, most recently Bob Heflick and colleagues in 2020. So you can read more about this situation. There are international agreements on germ cell testing. Most members of the United Nations ascribe to the Global Harmonization System, GHS, which is a method to classify the hazardous nature of chemicals. The GHS, at least on paper, does consider a human germ cell mutagen to be of equal alarming nature as an agent that would cause cancer or cause reproductive effects. But as usual, it does not provide a method for determining if an agent is a human germ cell mutagen. So I think this has been the real problem all along. REACH in Europe and the European Chemicals Agency, neither of these require germ cell testing, but they do note that or imply that if one finds an agent to be a somatic cell mutagen in the rodent, it should be considered a somatic cell, a germ cell mutagen as well. And this is ensconced in law here in the United States as well. I've put a quote from the FDA document here. I won't read it, but essentially, most agencies here in the US and worldwide ascribe to the notion, to the assumption, that if an agent is a somatic cell mutagen in the rodent, usually it's the mouse bone marrow micronucleus assay that's performed, one assumes it is also a germ cell mutagen. If it's a somatic cell mutagen, it's likely a germ cell mutagen in vivo. And conversely, if it is negative for somatic cell mutation in vivo, the assumption is that it's negative as a germ cell mutagen in vivo. So the question is, how legitimate is, or how valid is that assumption? And the answer is not very. So not all germ cell mutagens are detected in mouse somatic cell mutation assays. N-hydroxyacrylamide, cigarette smoke, various chemotherapeutic agents, all of these are negative in the mouse bone marrow micronucleus assay as somatic cell mutagens, but all of them are positive in mouse germ cell mutation assays. So a number of agents are indeed missed by the somatic cell assay. So what might the future of mutagenicity testing look like? Well, a number of scientists around the world have very recently, these are 3 papers, all 2020, have discussed this. And so I'm summarizing their views right here, which is that somatic and germ cell mutation should be assessed in a single animal, and that assay should be integrated into standard repeat dose toxicology studies. Mutation as an endpoint should eventually replace carcinogenicity studies, and in utero exposure should be assessed for genetic mosaicism, which currently is not. And of course, next-gen sequencing and changes in gene expression should be done in such germ cell assays. I should note the obvious, which to this day, epigenetic testing is not done at all. And I won't go through the details of all of this, but we know that mice exposed to air pollution and male humans exposed to ionizing radiation all can father pups or children that have changes in gene expression. So this is well understood. The next speaker will discuss this further, I am sure. And so it's an important phenomenon that can lead to rather bad outcomes in the children. And this kind of testing is not incorporated in the regulations at all. So let me give a conclusion and recommendations. So current testing regulations are insufficient for germ cell effects. Mutagenicity testing should, of course, include the Ames assay, but it also should include the mouse liver micronucleus assay. Currently, it's the mouse bone marrow micronucleus assay. The reason it should be the liver micronucleus assay is that there are studies showing that some germ cell mutagens that are missed by the mouse bone marrow micronucleus assay are picked up by the liver micronucleus assay. And that's because the liver has greater metabolic capability than bone marrow. So I think one would do much better to do this in the liver rather than the bone marrow. Mutation assay should be included and should be integrated into standard repeat dose tox studies. Standard gene expression assays in somatic and germ cells should be developed and included with mutagenicity testing, and mosaicism should be evaluated and included in reproductive tox assays. Of course, molecular epi studies should be done to assess mutations and gene expression changes in children and sperm of men exposed to smoking drugs and other environmental agents. This is the last slide. Obviously, an international agency should be empowered to evaluate agents for their ability to cause germ cell effects in humans. I would argue that it should be IARC. Funding agencies, of course, should be supporting research on germ cell effects in exposed male and female rodents and in humans. EPA in particular should reconsider its current plan, which is to reduce and possibly eliminate animal toxicity testing and replace it with high-throughput testing. And this will be very bad because as far as we can see into the future, animal testing will be needed to assess germ cell effects. So this would not be helpful. And then I would point out that the CompTox program at EPA and Tox 21 program at NIHS, which all feature a high throughput testing, none of these include assays for mutagenicity, none of them include assays directly for carcinogenicity, and none of them include assays for germ cell endpoint. So I would view all of these as insufficient for the safety evaluation of chemicals for human health effects, especially of future generations. Thank you very much.